Christy Carter today for her talk on the gut aging and interventions and implications for physical and cognitive decline. Uh, Dr. Cart, I personally know Christy from a couple of years. I met first time her in Wake Forest School of Medicine when I was there, and since then we have been in touch and trying to initiate some of the collaborative studies recently. And Dr. Carter is associate professor in University of Alabama, Birmingham, right?
Yes.
And she is expert in aging related studies in different models as well as in the human clinical cohorts. And her recent one of the area of interest is the microbiome in aging, and that's what she's going to talk to her today. And so let's welcome to Dr. Carter again, and Christy, floor is yours.
Thank you so much. I'm really happy to be here, and it's really cool to see familiar faces of people I haven't seen in a while, and people maybe I haven't fully met before, but have read their stuff, and so it's really a pleasure. Um, the title of my talk is a little bit vague. I was kind of alluding to that earlier. I know this is a diverse audience, and I'm certainly not a microbiologist, but I'm more of a person who is interested in studying aging and changes in cognitive and physical function. So that's where my interests primarily lie because that's, I think if we're not changing function with the interventions that we're doing, then not sure why we're doing the interventions that we're doing. And so I always like to have those outcomes. Recently, as Hariam said, that I've taken up an interest in the gut microbiome and how it impacts aging and physical function. And specifically, I'll talk more about cognitive function today, but also how that change to the gut impacts distal organs, which are involved in these processes, such as the brain and the skeletal muscle system. But that's just a big overview. I am at the Department of Medicine at UAB in the Division of Gerontology, Geriatrics and Palliative Care. We just got a large endowment or just received a large endowment. And so now we are known as the Hearsink School of Medicine. And it was kind of funny that when I was looking at the branding stuff today to get this picture, they actually even had a little phonetic of how to pronounce the name of our new School of Medicine. So Hearsink is the way that you say that. So that's a little bit about me. Let's see if I can. Whoops. I'm gonna. There we go. Now I've got it. So I like to thank the people at the beginning of the talk, because if I go a little too long or if I forget anybody, I feel really bad. So these are people that have helped me over the last few years get this work off the ground, and lots of different people who are in Florida at the Institute on Aging. Tom Buford, he is I call him my lab buddy. He and I have a joint lab together where I do a lot of the animal work, and we translate that work over to humans. So he's doing all the clinical work that the little bit of which I will tell you about today. Yi Soon and Lisa Roberts were both postdocs in the lab, and they really helped get all these data collected. They both have gone on since to jobs, actually both of them in the Boston area at small teaching colleges. So they're very happy. Then two really great undergraduates, Anthony Knighton and Sue Permsetti. Two great technicians, Anisha Banerjee and Yu Fong Yang, who spent a lot of time administering these drugs to our animals, which I'll tell you about in a minute. And then just all of our other collaborators for all the other things that we're doing with these models. Some of these data I will present, some I won't, but they're still contributing to the larger goal of the lab. Also, in particular, I'm going to present some fly data today, some Alzheimer's fly research data, and this was conducted by Dr. Patty Jumbo Lucioni at Samford University, which is a really a little small liberal arts college that's here in Birmingham, and she does amazing work. So I really wanted to give her her own slide because for being at this relatively small institution, she's really doing some big things. Okay, so by way of outline, I'm going to first talk to you about anti-aging interventions and why aging is a problem. Then we'll go through these other topics, primarily talking about the gut and inflammation, the kind of interventions we're using and why we use them. Proof of concept study, where we were looking at some of these genetically modified probiotics that I'll tell you about, and just to see if it would work in our animals. And then I relate it back to the gut-brain axis and some of the data that we collected to show the interaction between our interventions and the brain. And then I'll tell you, as I said before, about Patty and her fly studies. And if we have time, I'll talk to you about some of the multimodal interventions that we're doing, combining these GMPs with exercise, because that's another focus of our lab. We're interested in the effects of exercise on aging and all of these outcomes as well. And then I'll mention some follow-up studies we're doing, and then we'll be done. Okay, first, anti-aging interventions. So I wasn't sure if this group of people knew a lot about aging, given that it was a diverse group of people, but now I see that maybe you know more about this than I do. But I just like this slide to show people that the growth of the aging population, that it's getting older, and this is going to become a public health problem. When we're going to have 22% of the total worldwide population being over the age of 60, just the cost for healthcare alone and the social services that are going to be required to take care of these individuals, not only socially, but even family and caregivers, is enormous. So this is a problem that we need to deal with now. It is already here, and it's just going to keep getting bigger. So it's not like we can go back. I mean, it's great that we can go do prevention stuff, but we also need to do things right now that will help mitigate the decline in the health of the older population. And this was really made known with this 2016 report of key indicators of well-being in older Americans. And these are the number of adults. This was caught up in 2018. But basically what this shows is that the number of adults in the millions with these different types of disabilities. And one thing I like to point out is that the two, especially over the age of 65 here, what you see is cognition and mobility are two of the biggest areas for disability within this population. And you can see that 60% of these individuals over the age of 65 are going to have some kind of disability. So this is something, and this really brings down the quality of life. If you cannot ambulate in your own home space, if you cannot toilet yourself, if you cannot feed yourself, if you can't balance your checkbook, if you can't do these simple activities of daily living and kind of more complex activities of daily living, you can't remain independent in your own living space. And I think that's the number one concern of older people, is that they will not be able to be independent and will have to either go into assisted living facilities or depend on a caregiver. It's also become apparent that people see that there is money to be made from creating these anti-aging interventions. Several years ago, Google opened up the company Calico and actually recruited a bunch of top-notch biology of aging type longevity experts to begin working on compounds to extend longevity. And just recently, last month, Jeff Bezos, I guess after he conquered space, now he wants to conquer aging. He's supporting this Altos lab in Silicon Valley, which is where these are. And there's just also, in case you didn't know, these impetus grants that are being released by a group of entrepreneurs who are doing high turnaround reviews in as little as 2 weeks of applications that they will give $500,000 to individuals who are looking to develop anti-aging interventions. And I should say that most of these individuals focus on longevity or lifespan. But the idea is also that there could be a concomitant benefit to health span as well, meaning that we're not going to make people live to 120 years, except they're going to live with all the diseases they already live with as they age. So we're not going to make them live for an additional 50 years in a morbid state. But instead, as we are able to extend the lifespan of individuals, health span will follow. So that's the idea behind this. And for me, as I said, I'm a little bit more interested in health span at this point because my central goal is to improve quality of life in older people and make them able to perform for themselves and for society in general. So many of these startups are tackling what are called the hallmarks of aging. And so this paper came out in 2009, and it's been pretty influential, basically stating that there are 9 causes of aging that are biologically based. And if we can tackle these different causes of aging, then not only can we slow down the aging process, but the diseases that are related to the aging process. So it's taking an approach that we might be better served instead of trying to cure disease, that we cure aging, and then that will kind of get rid of all of these diseases. And so what people do is they target these various different hallmarks. So they might make knockouts of animals that have, for example, rapamycin is one of these huge compounds that has been known to slow aging in mice. So it involves the signaling of the mTOR pathway. So knocking out mTOR and then seeing what these animals look like. So that's kind of been the approach, and/or coming up with very specific pathways and targeting them pharmacologically, as these companies are doing to try to come up with these anti-aging interventions. So kind of central to many of these hallmarks of aging is the concept of inflammation. And I know Paula probably knows about this more than anybody sitting here. And so almost any dysregulation in any of these hallmarks in particular will lead to chronic inflammatory states. And that is a hallmark of aging in humans itself, across multiple species, that as these organisms age, chronic inflammation ensues along with that. So we started to think about this in terms of gut health, and I'll tell you a little bit about this part, then I'll tell you a little bit how we got interested in the gut. So maybe this is a little bit backwards. But if you look here, and this is from one of Tom, my lab partner's studies from several years ago. These data are old, but I think they still hold up. You'll see this trend keep going. If you look at the number of publications in PubMed when searching aging and inflammation, I mean, almost nothing like 30 years ago. And now what you can see is there are many, many papers. And like I say, this would probably be much bigger if I had renewed this graph. And it's been a question for years and years, where is this inflammation coming from with age? Like, why does everybody have, why is it so prevalent? And so Tom wrote this article to look at, and this was I should say that graph came from this article, to consider that maybe the gut is the source of this inflammation that we see with age. And I don't have to tell you guys about this again, but that there are many, many times more genes from microorganisms in our bodies than are in our own human genome. And these microorganisms are highly involved in numerous metabolic reactions that are going to influence our metabolism, our behavior, our physiology. And 70% of these lymphocytes reside in the gut and are associated with lymphoid tissue. So, which makes sense, because if you're bringing things into your body from the outside, the first place that they're going to hit is in your gut. And so being able to make sure that you can protect against those kind of bad bacteria versus good bacteria is really important. So we begin to think, well, if this is the case, if the gut becomes dysregulated with age, maybe the gut could be the source of some of this inflammation. And this is also why I recently wrote a paper talking about, is it appropriate maybe to make gut aging or gut dysbiosis the 10th hallmark of aging? So I'm just giving a plug for that paper. I think the jury is still out on that question. We're also going to have a discussion of this at the AGE conference next year. So hopefully those of you who can attend, hopefully that will be, we'll all be together for that one. So it'll be a really good discussion to have. So one of the reasons why we're going in this direction is because if you look at aging, as I said, it goes along with inflammation, and not only that, aging and inflammation are associated with dysregulated physiology, not just in the nervous system and the muscle system, which is what we study mostly, but almost every system that you can think of. And we know, and this, I'm just providing an example of one study, that chronic inflammation or markers of inflammation are associated with cognitive decline. And I'll show you later also that this is also the case for AD. So when I wrote this paper, the 10th hallmark of aging, we went back and looked at gut microbiota and disease, and looked over the last 20 years. And I mean, there was just a huge peak in the numbers of papers. And actually for aging, so that's like 5,000. That's huge. That field has grown from nothing over the, 0 papers over the last 20 years, up to 5,000 papers in 2020. We're lagging behind in terms of aging, but still the trajectory is the same with ever increasing number of papers. What we're thinking, so this is the exact same image that I showed you before, but instead of inflammation, we have here gut dysbiosis. And gut dysbiosis is also associated with dysregulation in all of these systems that I show here. Excuse me. It's also associated, and like those 5,000 papers, there are so many of them now, but in particular, if we're talking about frailty, it's associated with microbiota. And also, more recently, pathological markers of AD have been shown to be correlated with cognitive impairment and severity of pathology in patients with Alzheimer's disease. And there are now some papers even in some preclinical animal studies showing that probiotic administration may help to ameliorate AD, well, in AD models, mouse models, that is. So there seems to be this relationship between inflammation and gut dysbiosis that kind of layer upon each other and affect multiple systems, including the central nervous system. So this was a paper we published a few years ago, also showing that when we just looked in the serum of young and older individuals, and we looking at RNA-seq to see what the relative abundance of different types of bacteria were floating around in the serum, we saw that and we looked at many things, but I'm right here, I'm just showing you one example of one of the things that we found is that an increased abundance of Bacteroidetes in young adults relative to older adults. And we then correlated them with indices of chronic inflammation like IL-6 and TNF-alpha, and also a marker of relative health in older people, IGF-1. And what you can see is that there's a positive correlation with Bacteroidetes and IGF-1, and a negative one with IL-6 and TNF-alpha. So this was showing for one of the first times that we could actually measure the bacteria in the circulation and correlate it with circulating factors that contribute to inflammation and that are predictive of declining cognitive function and declining physical function. To be sure, given this, we could not tell if these were dead bacteria or live bacteria, but they were leaking out somehow and getting into the system. So that was pretty interesting. So as of now, then we decided, well, we want to be able to talk about this in a way that makes sense so we can go in and do interventions to mitigate this inflammation in the gut and see if we can change its relationship to functional outcomes such as cognitive measures and physical performance. So this is kind of what we were working with, is just this ideas that are risk factors for a dysregulated gut. We're particularly interested in biological age. With gut dysbiosis, two things can happen. You can just get a change in the composition of the microbiota, which causes, indirect inflammatory signals, signaling not the bacteria themselves, but inflammatory signals that can get into the circulation and cause chronic inflammation. But also just the bacterial microbes themselves can get in the circulation and cause that inflammation. And then this would reach distal organs or distal tissues and would cause aging and or age-related disease. So the idea is that if we can somehow impinge upon this, then we could mitigate this. So the first thing that we started to do was look at genetically modified probiotics and the renin-angiotensin system. So we published this paper last year. It was a review looking at just the gut microbiome as a therapeutic target for cognitive impairment. And this is just another way of looking at what I've just been telling you for the last 6 slides. But what it shows is that, again, there are these different causes of dysbiosis and different therapies that we can look at. And we are starting to look at all these, but I think today I just wanted to begin to talk to you about our genetically modified probiotics and why we're using them. So first, I want to talk to you a little bit about the angiotensin system, because what we've done is we've packaged this peptide into a bacteria so that it can be released into the gut and get into the circulation. Some of you may not know a lot about the renin-angiotensin system. If you know anything, you probably know that it's the major regulator of blood pressure in the body. For many years, it was thought that there was kind of only this one arm of the renin-angiotensin system, whereby Ang2 binds to AT1 receptor, and this is what's going to increase your blood pressure because it causes your vessels to vasoconstrict. But it also has been shown now that there are signaling pathways downstream of this receptor that are apoptotic, they're pro-inflammatory, and they promote lipid deposition. Then probably, gosh, I used to say 10 years ago, probably now like 20 years ago, this arm of the RAS was delineated, and it's the kind of the opposite and oppositional arm to the Ang2 arm, and this is the beneficial arm. And so angiotensin 1, depending on the enzyme that cleaves it, can either produce Ang2 or Ang17. If it produce this ACE2 cleaves and it produces Ang17, it binds to this MOS receptor, and it, as I said, it has all the opposite and beneficial effects. What's kind of interesting is that ACE2 localizes in enterocytes into the endothelium of vessels from the stomach and intestine and colon. And this has been really interesting because, as you all know, ACE2 is kind of the gatekeeper for the COVID, for the SARS-2 virus. And this has been one of the symptoms of COVID is to have gastrointestinal upset. So yes, as I said, this is the bad arm, this is the good arm. And so with age, what happens is there's an imbalance between these two, such that there's high levels of angiotensin 2 and low levels of angiotensin. So the idea, and so for years we've been trying to impact this side of things with different pharmaceuticals, but we only make minor. We've only had minor success in staving off some of these age-related conditions. But more recently, when we intervene on this arm, we have a much better effect. So it seems like the better strategy then is to target this arm and increase it rather than to try to decrease this arm. Okay. So it's not only that ACE2 is in the gut, but it's everywhere. And I think, again, it's going to be pretty interesting in the future to see what the implications, the idea of having these local systems everywhere in the body in terms of long COVID. I mean, I think there's some research going on out there regarding this. So it's everywhere. This is one of the first papers that got me kind of excited about thinking about the renin-angiotensin system and the gut. So this is a mouse model, and what they've done is they've given these hypertensive rats. I'm sorry, I said a mouse model. This is a rat model. So gave these hypertensive rats an ACE inhibitor, and then looked at their blood pressure and just the fibrotic area in these different areas of the gut. And what I probably just want to point out to you is if you look at everything in red, these are the hypertensive animals. And then if you look at all of these other bars that look the same to each other, they're either wild type animals or wild type treated with ACE inhibitors, or hypertensive animals treated with ACE inhibitors. And basically what happens is these ACE inhibitors returned arterial function, mean arterial pressure to normal in these animals. Oops. And that there was an improvement in gut pathology that was noticed in these hypertensive animals, and that ACE inhibitors reversed it. So that was like, wow, that's kind of incredible that you can give an antihypertensive medication to a hypertensive animal and show these improvements in the gut. So what might be going on there? This was another paper by Mohan Razada. I'm not sure if you guys know him. He is at the University of Florida too. So it's just another study that showed if you induce hypoxia in either wild type or ACE2 knock-in mice. So these would be mice that have overexpressed ACE2, which increased levels angiotensin. You see some of the same patterns, that you have this profound effect on changes to the gut. So these two studies were in terms of hypertension. So I think that's really where the field began to really understand the relationship between the renin-angiotensin system and the gut. So for our current study then, we were super interested because many of so these are age-related diseases. And so if we can have an impact on these types of age-related diseases, then maybe we could have an impact on aging, which would also mean functional aging, which is what I'm interested in. And so we came upon doing this completely by accident. I was really didn't have a lot of experience with looking at the gut for anything. And so I went to Mohan and I said, you've had all these really cool findings, like how are you what are you doing now? Like, how are you doing your interventions? And so he was doing two approaches. One was he was taking tobacco leaves and cloning the human, either ACE2 or Ang17. And these tobacco leaves were basically releasing them into the gut. And it was fused with this CTB element, which then would cleave it and let it be kind of by lysosomal action, it would get into the circulation. So he was raising Ang17 and ACE2 levels this way. And we had written a grant to do it this way, but it turns out that it takes so much plant material to feed the rats in order to get them to increase the Ang17 levels that it wasn't practical. And so the next best thing was to instead put it into these to Lactobacillus paracasei. So that's what they started doing. And it was great because what they could do is they could make it at UF, they could lyophilize it and freeze it and send it to us, and then we could reanimate it and we could give it to our rats. And the other great thing about this is that we could also give very precise, localized amounts to our rats, because we decided we would gavage them with a compound so we could know exactly what they were getting. So the first study, so this was a way that we thought we could manipulate Ang17. In the literature, it's very hard to just inject Ang17 unless you're using or get it in the circulation and get it to stick around, because it has such a short half-life. So a lot of people will just use osmotic mini pumps, or they'll do direct infusion into the brain. But we wanted to make sure that we were doing something that was more translational, that this was something that could potentially be translated to humans, because you're not going to get people wearing pumps of Ang17, and I'm pretty sure you're not going to get people to walk around with cannula in their head, infusing Ang17 into their ventricles. So we thought this is an improvement over what's been done in the past, and it could have the benefit not only of increasing circulating levels of Ang17, but also targeting the gut and potentially improving the leaky gut and dysbiosis as well. So it would be something that packed a punch in two ways. So let's see if this works. What we did is we took these 24-old Fisher 344 brown Norway rats, and we had 29 of them. And this is just to show you that they do pretty well with gavage. I think a lot of people were really, that we got criticized initially that the rats wouldn't be able to take it. And it's a pretty high volume that we're giving them as well. But as you can see, they took to, I mean, you had to habituate them. They weren't happy right at the beginning when we gave it to them. Anyway, we orally gavaged them for 4 weeks as proof of concept. These were all male rats. And then we gave it to them the same amount of bacteria expressing or releasing the Ang17 0, 1, 3, or 7 times a week. And we collected blood. And so we just wanted to see if we could raise Ang17 levels. So what is important here is that over that time, we looked at their food intake and we didn't see any difference in their food intake. So it's not like the bacteria or having some extra volume in their stomach did anything to their food intake, and it didn't do anything to their body weight either. What was very cool is that we found, and we also did a systemic injection just for comparison to see what these levels look like. And what we found is that 3 times a week really seemed, if we're doing with the oral gavage, raised levels of Ang17, really the best, lowered levels of Ang2. So we were actually able to control both arms of the RAS with this compound. And the same thing with the systemic. But then we looked at dysbiosis in these animals. What we saw is with the oral gavage is that the 3 times a week was really the one that we got the best results from in terms of diversity and richness. We had a lot of There was just a lot. These data were much tighter in the oral administration, and there was really no difference at all across the systemic groups. One of the things, it's not going to do my animation for me. Oh, one of the things we showed, anyway, I can just tell you that one of the increases in abundance was in Akkermansia, uniciphilia, and we saw that in the 3 times a week group that we had the greatest relative abundance. And I'm sure most of you also know that there is a protective effect of this bacteria in a mouse model of Alzheimer's disease, and that there are some correlational studies in humans showing that changes or the amount of this bacteria correlates with amyloid load. So when we looked at the prefrontal cortex in these animals, again, what we saw is in 1 to 3 measures of our inflammatory cytokines in the prefrontal cortex itself, especially at 3 times a week, we saw a reduction of bad cytokines, and we saw an increase, a reduction in all these dilatory cytokines. So our conclusion was that, yeah, we can do this. It seems to work. We increase levels of Angiotensin. We see an anti-inflammatory effect in the prefrontal cortex, and we see a change in beta diversity and abundance in the gut. So we think this is going to be highly translatable. The cost of the production is extremely inexpensive, and it's just a decreased burden for regulatory approval as well. So what we don't know right now is how all of this is happening. And so it could be one of 2 ways. One is it could just be that the Ang-1,7 is actually getting into the brain, although it seems kind of unlikely because it's such a large molecule, and that it works at these various receptors that I showed you before. That all have, so this is where Ang2 would normally bind, so it has all of these deleterious effects in terms of oxidative stress damage and accumulation of tau and A beta. These 2 receptors are kind of good receptors. MOS, Ang-1,7 binds to MOS, which has a direct action on this AT2 receptor. So it could be that that's how it's happening, is that Ang-1,7 is in the circulation, it's getting into the brain, and it's having these effects. But it could also be that there's some direct effect of signaling from the gut to the brain independent of the Ang-1,7, that somehow Ang-1,7 is changing what's going on in the gut and influencing the brain that way. And here are multiple ways that could happen. Tryptophan metabolism is an interesting target, and I don't want to scare you with this big flow chart that we made, but the metabolites of tryptophan, if you see in the red or you see in the green, can either be neurotoxic or they can be neuroprotective. And there are many places along this pathway. So for example, here Ang-1,7 has been known to signal dysfunctional or lack of it, tryptophan absorption. So anyway, this is kind of a complicated scheme of what all those metabolites look like. But one of the things that we have shown with these animals who were dosed for 4 weeks is that we do see changes, and these are small changes, but they're significant, in 3 times a week in serotonin and phenylatic acid, which is part of a neuroprotective pathway for tryptophan signaling. And so we're trying to look more into this and see if this is and to get at exactly what these mechanisms are. Are they truly systemic, or are they based on changes in gut indirectly relating to changes in signaling to the brain? And this also came from some of the work that we did with Patty, as I said before, this fly dysbiosis project. She's got a neat system set up where she can do cognitive testing in flies, and so she got this strain of AD fly, and she let them grow up to be 14 days of age, which is pretty old for a fly in this case. And what she did was, so flies enjoy being in the light. And so with this assay, what you do is you get the flies and you get them kind of in this loading chamber here, and you let them go into the light. And then what happens is you blast them with a very noxious smell, and they do not like it at all. So then when you go to test them, they associate this noxious smell with the area of the light. And so now when you allow them in this chamber, they prefer the dark. So it goes against their kind of evolutionary preference for light. There's a lot of stuff here. I just want to orient you to this. We gave them we call it the LPA, the genetically modified probiotic. So if you look here at trained, these are females here and males here. So learning in this case will look like the numbers will be increased. So if you look here, the trained control animals performed very well. They preferred the dark much more than they did the light. But when we gave the intervention to the AD animals, there was no impact in the females on their cognitive function. But when we gave it to the males, which are seen here, there was an improvement in their cognitive function. So there's a sex-specific effect of this, the effects of not only learning here, but also on the effect of the intervention, which is interesting because it is the case that more females get AD than males. So not sure exactly what that says and translate all the way up to flies. Anyway, this kind of provides us with more of a high throughput methodology for looking at different combinations of bacteria and different components of the RAS, which we're writing a grant on that right now. We also were able to show, and we're still collecting these data, that in the AD animals, kynurenine, which is on this kind of neurotoxic pathway, is highly elevated. It's barely detectable in the control animals, but it's highly elevated here in the AD animals. So again, this provided us with some kind of initial idea that this is the pathway we should be looking at. So it's nice to be able to have this model in order to inform what's going on with our animals. Now, we were looking at just normal aging, but we do have a rat model of Alzheimer's disease that we're working with right now and looking at these changes as well. So nice use of the fly in Patty's lab to help us inform what to do in the rats. The last thing I'll talk about that we're doing is we're looking at multimodal interventions, combining exercise with trying to modulate these different areas of the renin-angiotensin system. And these are some data from my lab looking at male rats who we gave enalapril, which is helps to lower the levels of Ang2, that bad RAS compound. And so this is treadmill endurance in animals that have been given just enalapril alone, exercise training, or the two together. And what we saw was kind of this not very synergistic, but additive effect of enalapril and exercise together. And this is really where Tom and I started working together the first time, because he took a data set of a bunch of older adults, over 3,000, who had done exercise training, who had information about all of their medication status. So we were able to look at individuals, their gait speed after they had been in this exercise program for 4 years, and their short physical performance battery. And in particular, we were looking at ACE inhibitor users. And what you can see is they're changing gait speed. This was the only group compared to everybody in the whole study, and or people that used a different type of antihypertensive medication. So this was our first indication, like, wow, that's pretty powerful. And then when we also looked at these different circulating inflammation markers, again, bringing the story back to inflammation as kind of an underlying mechanism, we did see that the ACE users had less inflammation in the case of these two markers than their counterparts. So this is some data that we have just collected. And these are my last couple of slides. And what it shows is, again, sex differences between males and females and exhaustion time. So these are sedentary, these are exercised animals. And this, if you look at the red, these are the animals who've received the genetically modified probiotic. And this time we were able to compare it to the probiotic as well. And what you can see is that in the males, exercise helped, but there was no added benefit of the LPA. Of the genetically modified probiotic. But in our females, we did see that benefit. So again, that's very interesting. However, when we looked at cognitive outcomes, we did a novel object recognition with these guys. So it's just, you show an animal, get it used to an environment, show it a couple of objects, it gets used to those objects, and then the next day you change one out and it's like, oh, that's different, I'm going to pay attention to that one. And so it's some indication of hippocampal and prefrontal cortex function. Anyway, there was no difference in the males with exercise or with the treatment with probiotic or the LPA, but we did see in the females that this combination of the exercise and the LPA did improve or did mitigate the decline in novel object recognition. Okay, so all of our follow-up studies, we're going back in all these tissues, brain, gut, liver, even to look at RNA-seq, metabolomics, metagenomics. We're going deeper into the sex differences between these groups of animals. And as I said, we're also going to start looking at our transgenic AD models to see if we see similar changes. We also have a pilot with some folks to look at this genetically modified probiotic in a non-human primate model of hypertension. Really interesting findings of the relationship between leaky gut and hypertension, leaky gut preceding the hypertension, and see if we can either reverse or prevent these effects. So we're translating across multiple animal models, I guess, and hopefully the ultimate animal, humans, that we can get this into humans. Thank you. Wow, that ended abruptly. Sorry. I packed a lot in there. I was trying to cover a lot of the things we've been doing. I went very fast. I'm happy to take questions. And I just want to show you this. This is Alabama. These are real mountains. And this was one of my surprising shots that I took on a bike ride through the mountains. And so I just want to invite you all to come visit one day when this is all over and come see UAB. Thank you.
Oh, beautiful. Thanks, Christy. Wonderful talk, wonderful work. Great.
Thank you.
Any questions? So I think so. I will just start with very kind of my own curiosity. You mentioned about the dysbiosis. Do you use that term into the grants?
Yes and no. It depends on how we're targeting it and who we're targeting it to. But yeah, I would like to hear your critique of that.
Most of the time, like, I think so. My feeling is people don't like it, especially in the grants. I mean, the papers, I think so. So, but I wanted to hear like whether you got any kind of feedback for that way.
We haven't, because we've been targeting a lot of the NIA study sections. So I think there has been less. Now so one of the grants that we submitted was with the flies, was to like repackage various components of the renin-angiotensin system with different types of bacteria. And we were calling it dysbiosis, and it went to a hardcore gut study section, and it just got trashed. It was, I'll have to, yeah, maybe you should, we want to resubmit that. Maybe I should get you on that one, because it was, yeah, we were not using dysbiosis correctly, and or it's like, what is it? What are you actually defining as dysbiosis? Is it that loss of diversity, richness, or is it actually the leaky gut? Is it the lack of diversity is causing the leaky gut, and then that whole thing together is what it is? And so, yeah, that's why definitely going to those study sections, you can't be vague in any way, shape, or form about what these things mean. But also inflammation can be a very vague thing, right? You
Yes.
can see like all of those different potential pathways and then you get the inflammation in the brain. What does that mean, right? Is it the microglia? Is it the neuron? Is it, are you messing up connections? Are you getting rid of cells? So that's why sometimes this is so hard to present, and it's also so hard to write a grant on, because like having it be at the really hardcore level at each of these things is a challenge. So we're expanding our team to make sure that we have those areas covered now, so that it looks much.
Another general question, because we were talking about the flies. Does the fly has the lactobacilli? Has anybody has done the whole microbiome sequencing on the flies?
It doesn't have it, but it gets in there. And so we've been one of the things that we're going back now and doing. So you have to collect so many little fly heads to see what you have. So we're going through and we're having to use mass spec to look at of course, for the tryptophan stuff, but also we're developing using mass spec to look for Angiotensin specifically with the flies, so that we can get those measurements, because you can't get enough flies to just do an ELISA on these things, for sure. It does not work whatsoever. So yeah, we're trying to improve that model that way as well.
Hariam, I have a question. So first, I apologize because I could only attend to a part of your presentation. So maybe the point that the question is maybe absolutely irrelevant, and I apologize in advance. But no, there have been a lot of the conversations regarding when you use probiotics as to whether or not they really colonize the gut, and all the papers from Dr. Segal on the fate of the probiotics. And maybe you have already touched on this, but what is really the fate of what you are using in the gut? I understand that you are using a specific way to deliver, the probiotic. I do understand this, but have you investigated what exactly happens to these molecules?
It's a great question, and that is one of the things we have not done. And so we really have to. So our collaborators have done this in the context of looking at diabetes and delivering these genetically modified probiotics. And so they've looked in the colon and like fluoresced the bacteria and have been able to see it. We have to do that in our rats, and we have not done that yet. So that would be another next step, because really, I do understand that it's important where these bacteria are colonizing, right? So if they're in the lumen versus if they're in the epithelium, the L cells have lots of ACE2 there, and Hariam and I have talked about this, and they interact with GLP-1. So I think that would be great to be able to show that it's there. And that it's having this influence on metabolism that way, because I think that's one of the mechanisms also that might be influencing this inflammation. The only way we know right now that it's around is that we have increased levels of this Angiotensin, and doing it 3 times a week keeps the levels high. If we just stopped all of the sudden, at the end of that study, it was 4 weeks, so we had to get rid of the animals. But yeah, it would be cool to see how long, if you take them off of it, how long it stays around. But I think the fluorescent bacteria studies would go a long way in answering that question for us.
Thank you, and congratulations for your talk. Thank you.
Well, thank you for being here.
And any more questions someone has? So, Christy, I just kind of one following question is, does these bacteria secrete the Ang-17 or just when they die, then it get released?
So it releases it. So it has that CTB unit, which is like a lysosome. So it kind of fuses with the this is, I'm going to butcher this. It fuses with the epithelium, and then it will release it out into the circulation, almost like a lysosome. And so that was put in there specifically to get it in the circulation, because again, this was all initially all about just getting Ang-17 out there, because all the other methods were not doing it in a sustained way.
So, Christy, it's Paula. I have a related question. I mean,
yeah.
So you have two effects, right? You have the Ang-17 effect, potentially.
Mm-hmm.
Have the lactobacillus effect.
Mm-hmm.
Can you separate them as to what's doing what?
Yes. So in that, I may have gone through it pretty quickly.
One slide where you showed a little bit of that, right? But yeah, did you show that for the cognition? I couldn't remember.
We did. Let me see. I always put extra slides in here. Can you still see that?
Yeah.
Oh, okay. Here. So this is the control, the gray. This is the LP. So that's just the probiotic alone. And this is the probiotic that releases the Ang-17. And so in males, and then of course we had exercise thrown in here too, which humans, it seems like human studies, people who study exercise like to throw everything, like the whole kitchen sink at this stuff. Anyway, this was really the only place where we saw an improvement, and that was with the combination of the genetically modified probiotic releasing Ang-17 versus any other group. So the probiotic alone did not do it.
But in the stuff that the studies from earlier when you were looking at the chemokines.
Yeah, so that was we did that one. We did not do the LP. We didn't do the probiotic alone in that study.
Okay, so I think that was where my question was.
Yes, we didn't have it there. Yeah. So now we are looking at doing all the RNA-seq in brains, everything. We're looking for everything. Paula, the other thing you might appreciate is now we've got a really complex maze set up so that we're looking at really higher order cognitive.
Yeah, I was going to ask about that too, but I figured you were eventually.
Yeah, yeah. But yeah, we understand it's not the best, but we were doing this whole battery of things. And so that's another next step. It's a biconditional discrimination task. And so, we've already found that we can train our AD animals to do some of it. They're not, they can do the simple object discrimination, but they cannot do the complex discrimination. It takes them many, many more trials in order to even get it. And so we have those data now. We just put them in our recent grant, and we're going to start looking at the intervention.
Great.
In that. But yeah, we should talk at some time about, looking at some additional inflammatory markers that you do, because I think that would be incredibly informative to what we're actually impacting. We are doing some work with Sarah Burke and Jen Bison. We got a McKnight grant to look at different types of interventions that impact the gut. So we're looking at vagal nerve stimulation, we're looking at ketogenic diets, and we're looking at our GMP.
Yeah, we can talk about that sometime.
Yeah, that would be awesome.
Great talk. Thanks.
Thank you. Sorry it was so fast and so much.
I have a quick question.
Mhm.
Do you ever look at C-reactive protein as a marker of inflammation?
So we haven't in this study, but we have in our human studies. And yep, I agree. Sometimes in the rat, the kits that they have for certain markers don't work as well as they do in mice. CRP has been one of those for us. And like if you look at ELISA, and it's also the case that some of the leaky gut ELISAs did not work like IFAB, didn't really work for us. And so that's why we said, okay, we're just gonna look at everything with metabolomics, with sequencing and see what we can find. And then we're gonna follow up with just some protein and mRNA studies as well. And I'll mention one last thing. We also did some lipidomics with Tim Garrett at UF, and because I'm also interested in the physical function side of this, and we did see that we see big differences in these combination groups with regards to triglycerides, diacylglycerols, and so that's another story about quality and inflammation in the muscle as well.
Okay, thank you.
Thank you.
Great. Anyone else? That's really fascinating work, Christy. Thanks a lot for.
Well, thanks for inviting me, and thanks for everybody for showing up and sticking around. Appreciate it.
Thanks a lot. Thanks everyone, and it's wonderful. Let's plan our following meeting, Christy, and we'll
Absolutely.
talk more on the community as well as the GLP-1 project.
I love that.
Great.
Sounds good. Okay.
Thank you.
Thank
Bye.
you, guys. Bye-bye.
Bye, everyone.
Bye.
Bye-bye, thank you.